Everybody
Uh,
got their paper up. Continuous fermentation by immobilized brewer's yeast.
yeah, we're not gonna jump right into that. We're gonna did that work? I don't know. My screen doesn't look right. Can you see that?
I see black.
Hmm.
Oh, that's darkness.
Oh, there we go. Let's try that one. Well, this sucks.
You can see your mouse.
I know.
That's it. I mean, that's a step up.
Not really. Technical difficulties.
Does anyone have a problem? Just, I mean, the paper is only 3 pages long, so it's not like there's a lot of room to get lost here. So if everybody can just open another window and look at the paper, we can get going.
Actually, 2 pages in a big fat black and white picture.
Mm.
Try
No, but
swapping
I
the
actually enjoyed that.
screens around.
I give it a bit hell, but it was enjoyable
Yeah.
because the architecture of the paper is sweet.
Mm. I like it because it's just they're like, here's the thing, it does the thing, ta-da!
Right.
I was
Exactly.
like, cool.
Very weird. Okay, I had to share it by screen. That's weird. Alrighty. So let me kick this off. Today I'm going to talk a little bit about a couple of things. Primarily bioreactors, since we're trying to make a bioreactor for Nigger. So, uh, oh, wrong window. What's a bioreactor, you might ask? Well, the commercial definition, it's an apparatus in which a biological reaction or process is carried out, especially on an industrial scale. And if you Google it, that's pretty much the definition you're going to find on everybody that wants to sell you a bioreactor site. The dictionary definition is a slightly different. It's a device or apparatus in which living organisms, and especially bacteria, synthesize useful substances such as interferon or break down harmful ones, as in sewage. There's a lot of sewage bioreactors out there. This is my definition. A reusable, contained, organized system of biological components that's designed to create a desired byproduct by biological, enzymatic, or chemical processes from some raw material. In particular, today we're going to be talking about yeast in the context of fermentation. Yeast, fungi, ascomycota, I practice these words, Saccharomycotina, specifically Saccharomyces cerevisiae, which is brewer's yeast. Brewer's yeast is a budding yeast. It reproduces asexually through a process called asymmetrical mitosis. Typically, what will happen if conditions are favorable, the mother cell will bud and produce a daughter cell, and then once a portion of the nucleus is transferred over to the daughter cell, it will undergo mitosis, and you'll end up with a slightly smaller daughter cell than your mother cell. It's a process that's aerobic, meaning that it needs oxygen. So if O2 is abundant and everything else is in line, it will go into this reproduction growth phase. Sugar is converted to energy, and then it uses that energy to reproduce and build the colony up. Another phase that yeast can go through is anaerobic fermentation, which is the one most people are interested in. If at some point the colony is deprived of O2, the yeast begins to harvest the O2 from the sugar and produce ethanol. Yeah. This is called anaerobic cellular respiration. In humans, the same thing happens when you overload your muscles, except instead of making ethanol, your muscles make lactic acid. When the sugar runs out or the temperature drops, the yeast go to sleep. They become dormant, and then they begin to flocculate. There's two types of yeast. There's flocculent yeast and there's non-flocculent yeast. Flocculent yeast will gather together in little groups, very social, stick together. The cell walls will actually stick together and form what we typically call yeast rafts in top fermenting yeast, or floaties in bottom fermenting yeast. This is an example of the two different kinds of beer you can get using just a different strain of yeast. It's the exact same wort. The one on the left was fermented with a highly flocculating yeast. So when the sugar ran out, the yeast crashed down to the bottom because they form these little like globs, and there's more mass in those globs, so they tend to separate out more quickly than the one on the right, which is a non-flocculent strain. Why is this important? Okay, well, if you're this guy, if you're the guy that's brewing, you know, hundreds or thousands or tens of thousands of barrels of beer, having a flocculent strain of yeast in your fermenter is a good thing. You'll notice that the fermenters are conically shaped at the bottom, very much like a separation flask. And the reason for that is so when the yeast is done doing its job, they all go to sleep, they flocculate out and settle down on the bottom. Then the brewer can then pump the finished beer out of the tank, the yeast stays behind, and then they have a choice at that point where they can either drop the yeast out of the tank or they can just pump in fresh wort. Okay, now we're on to the paper. Let's see if we can. Okay, so here's our paper. There is not a lot in this paper. It's a 3-page paper, really 2 pages if you consider the rather large, unknowable picture that they placed in here. But it condenses down to just a few key points, and that's one of the things I really liked about it. So it's a really approachable paper. Basically, in the introduction, they go through the process of explaining why flocculation is important and what they're trying to do with the paper. One of the qualities that you get from yeast is flavor profiles. So some non-flocculent yeast will produce flavor profiles that might be desirable in your beer. Therefore, you would want to use that non-flocculent yeast to make your beer. However, it doesn't scale up industrially because the yeast won't separate out readily. And there are other ways to do this. You can use centrifuges and all that other stuff, but that's expensive, and I digress. Main points here. I had to do a little bit of parsing because this paper is weirdly worded. But essentially, this comes down to alginate comes from seaweed. It's broken down with sodium carbide, and that yields sodium alginate. And it can be easily cross-linked with calcium chloride to form an insoluble calcium alginate. So you can directly mix sodium alginate into your beer, but you can't mix calcium alginate into your beer. Let's see. This section here basically says, you know, that they did this, that, and the other, and various yeast alginate ratios were tried, from 5 parts alginate, or excuse me, 5 parts yeast to 1 part alginate, up to 200 parts yeast to 1 part alginate. The concentration in alginate affected the resulting yeast flock size and shape, so they could actually, by varying the ratio, the density of that alginate gel that forms, they could somewhat reliably get flocks of different size. We go down here. I guess we're in the methods section now. Basically, this is the part that I had to actually copy and paste this out and then reparse the sentence properly. So basically, a solution of 1%. This is their procedure. A solution of 1% sodium alginate to beer by weight, mixed with a slurry of yeast at a ratio of 20 to 1 to 40 to 1 yeast to alginate, which comes out to about 2.5 to 5% alginate. The yeast alginate mixture was pumped into a solution of 10% calcium chloride in beer by weight. And yay, it worked. Basically, it formed a plug of basically a plug of somewhat semi-solid yeast that could basically float around the tank and act on the wort that was in the tank and produce ethanol, which is the end goal. And then this bottom part here is the analytics. And there was a couple interesting things I took away from this. So the health of the yeast colony was pretty well sustained over a 7-month period of reusing that same yeast. This isn't necessarily true in at least from my experience in batch fermentation, where you harvest the yeast and then repitch it in your next batch. Because the yeast are spending a lot of time in a dormant state. And that leads to unhappy yeast and yeast death. But by keeping the yeast fed, the colony seemed to be healthy for at least 7 months, which is a really long period of time. That's like 14 batches of beer. The other thing that I found that was kind of interesting, they talked down here about it was feared the immobilized yeast particles might also retain bacteria. So when you're producing beer, one of the tenets to making good beer is to sanitize and clean everything thoroughly, because you don't want a bacterial infection. Bacterial infections give you really bad off flavors. They can make you sick. But they actually took an infected batch of wort and ran it through the fermenter, and subsequent batches did not show any sign of infection. So the alginate did not pick up the bacterial infection, and the yeast in there. It's likely that the environment that the yeast has surrounded itself in there is a bit acidic and not necessarily something that bacteria wants to jump into. So, ooh, ooh. Okay. So why is all that good? Well, the yeast can stay in the fermenter. Once the beer is fermented, it can be pumped out the top, and fresh wort can be pumped in. The fermentation will happen faster and produce more ethanol because you don't have to go through the aerobic growth phase, or you will have a very short growth phase while the wort is depleted of O2. The fermenter does not require cleaning between each batch. SIP, or clean in place protocols, on a large fermenter can take hours, require lots of water, manpower, and caustic chemicals. So by using this process, the fermenter is no longer a one-shot use. It's now a bioreactor. Maker. Like maker with an N. We talked about this in our first journal club. You can see here some micrographs that I've got that show the structure. It's what makes up the hard outer shell of pearls, the inner layer of a mollusk shell. And this micrograph over here I thought was particularly interesting. It shows the area where the nacre is actually formed. You can see these little tendrils here. These are the proteins getting extruded out onto the inside of the shell, and you can see the layers, the varying layers of calcium forming within that matrix. Basically, this is just the structure of the mollusk shell. All the interesting stuff takes place in this boy, I hate that word, extrapital space right here, where the organism actually contacts the inside of the shell. Nacre's 95% calcium carbonate in the form of aragonite. I know we've already covered all this. And a 5% protein matrix in the form of chitin. I did find so I kind of went down this line of what do you call it, inquiry, where I started thinking about, so how does a mollusk do this, right? How does a mollusk produce nacre? And I did find a paper. It's mollusk shell, where you can find it in the library. Mollusk shell formation, a source of new concepts for understanding biomineralization process. The silk, basically, this is what they laid out in their paper. Their mineralization scenario. The silk phase is a gel that prefills the space to be mineralized. The chitin is ordered structural phase that ultimately dictates the orientation of the mature crystals. The matrix components are spatially differentiated. The first formed mineral is transient colloidal amorphous calcium carbonate, ACC. Nucleation occurs on the matrix, and the crystal grows at the expense of the ACC phase. During this growth phase, some of the acidic proteins are occluded into the crystal. I couldn't find the other paper that I really wanted to reference here. It might just have been an article and not necessarily a forum paper. But I started looking at the acidic side of this, and I discovered that mollusks, the acidic side of this, it's actually uric acid. So I guess it can be said that mollusks typically will pee into their own mouths because bivalves. What are you going to do?
Been wrong with the if you look in the voice and video chat link, I posted a picture of what I'm seeing, and it hasn't gone anywhere from the aerobic reproduction
Oh, Lordy.
diet. Yeah, I'm sorry to be interrupting,
Oh,
but
no,
I
you're
had to.
no.
I wasn't sure if that was just me, and I didn't want to interrupt because I thought everyone
Yeah,
else
every
was fine
single
with
one of
that.
us is feeling the same thing right now.
Wait, hold on. Screens. Is that better?
So
Looks better
far,
to
it
me.
looks okay.
Do you guys want me to roll back? I don't even know if that'll. No.
Now it seems to work.
Yeah, it works now.
So you can see the screen changing because I don't see that on my end.
I
Yeah,
can see
we
that
see
on
it.
my end.
Okay. So well, actually, I'm at the last slide. So can everybody see the little crappy drawing that I made?
It's not crappy, but yes, we can see the drawing.
Okay, so this was so when I got to thinking about, how do you make a bioreactor to make nacre? I started thinking about, well, what if you could make your own mollusk, like a synthetic mollusk? In the first paper, we talked about having a couple different bacteria that would one would put out the protein and the other would put out the calcium ions. And I started thinking, well, what if in a bioreactor the main thing is you don't want to flush your organism with your product. You want to get your product out and leave the organism in there to do the next batch of work. So I started thinking, well, maybe you could make an alginate SCOBY with the bacteria, and then cover your mold with that SCOBY and have it just kind of act like a mollusk does. And then, of course, the liquid would be nutrients and I'm sure minerals for the organisms to feast on and do their little dirty work. So, and that's pretty much all I've got, and that's kind of where I wanted to leave off today and chit chat about, because I'm sure Gabe has ideas.
Cool. First question.
Yeah.
So on I agree. The paper is kind of oddly worded, but just to double check, from my understanding, their ratios was what was it, like about 2.5% of alginate?
Yes, 2.5 to 5%.
2.5 to 5% alginate. Okay. And then the race, and that was for the calcium, that was that calcium alginate gel, or the sodium alginate, or was that, is that ratio applying for both?
Okay, so essentially it's two and a, we'll just go with the 2.5%. 2.5% alginate with whatever thing you're going to put in it, either yeast, bacteria, uh, pickle juice. Okay, this is also used in molecular gastronomy, as most of you probably already know. So it's a 2.5% solution of alginate and whatever you're putting in the alginate. And then that mixture goes into a 10% solution of calcium chloride. And that calcium chloride swaps, uh, the, how do I put this? The sodium and the calcium make sodium chloride,
Right.
right? And then what that leaves you with is the alginate, and it wants that calcium ion to attach to, and that's basically what cross links it, and it forms an insoluble shell. And the longer you leave it in there, since it's somewhat permeable, the longer you leave it in there, the thicker the shell gets.
Cool. Okay, second question. So the researchers were able to use this alginate fermenting gel for up to 7 months. How would the flavor impact from month 1 to month 7 of the beer, or would it change? Would it not change drastically?
Well, that's actually what they were measuring in this table, the analytics of beers from tower fermentation containing immobilized yeast compared to blah blah, are flavor volatiles. So diacetyl. Okay, so diacetyl is actually a byproduct of fermentation you don't want. So you really want that to be a low number. Um,
Okay.
that's like a buttery flavor, right? It's I mean, if you're making butter beer, I guess that's something you might want. It's okay in some beers, some beer styles, but generally it's something you don't want. Um, and propanol is it's more of an aroma component. Um, but your flavor sense is tied to your nose, so I mean it comes across as a flavor and you can see like it was pretty steady across the 7 months.
So my understanding is that flavoring can be like super concentrated. For example, I think there's a flavoring for artificial red pepper that, can make an Olympic-sized swimming pool taste like red pepper. But some other flavorings are, require a lot of volume in order.
Yes.
So in these examples, N-propanol, isobutanol, the the change I'm seeing, which is like plus or minus, 2 to 4 milligrams
Yeah.
per liter, that's not enough to drastically impact the beer taste.
um with some compounds that could be true, but you see, this is the thing, as long as it's steady over that 7 month period, you can make adjustments in your process, like
Okay.
when you're brewing beer. So I don't know how well versed you are in beer, but Belgian style ales have a very fruity aroma and flavor to them typically, and that's because the yeast throws off a lot of esters. You can do this without a Belgian style yeast by using, say, a lager yeast, and just stressing the yeast a little bit. Belgian style yeast don't need to necessarily be stressed to throw off those esters, but you can get
Okay.
other yeast to express different compounds depending on the environment that you give them, and that can be through changing the nutrients, that can be through changing the temperature, that can be through agitation, um, temperature cycling. There's a lot of there's if you ever get into brewing beer,
Mhm.
you'll have like 2 years of doing nothing but reading papers because there's so many papers written about beer. It's
Yeah.
insane.
So to summarize, regardless of this, this small fluctuation, brewmasters and brewer technicians can account for this.
Sure, I would
Cool.
I would think so. There might be a compound or two that you would, but I don't think. I mean, there is difference. That's not to say
Right.
that there isn't a difference because they've got the levels from a normal beer.
It's a living
Um,
thing. Yeah.
right, yeah. But if you look at you look at any batch of beer, no two batches of beer are the same. The only way that you get a Budweiser to taste like the last Budweiser you had is because they do it on such a massive scale that all the little variables like the barley that goes into it or the rice or the different genetic defects in this batch of yeast versus that batch of yeast, it all averages out when you're making 100,000 barrels of it.
Cool. Also, your screencast has frozen again.
Joy.
But thank you.
And
Oh
since
wow,
there is
that does a really crazy thing, doesn't it?
I would a little trick is also to disable all the people joining and leaving sounds, since it's a common thing to refresh the Discord when it bumps you out of the voice chat without telling you. It says you're connected and all, but the sound will disappear, and that the only way to fix it is to leaving and coming back. So yeah, sorry, take it away.
Is that a setting somewhere where you can disable that sound, or
Yeah,
it's not?
yeah.
I don't see it. Gain control. Try turning that on.
All right, so I think your screen's good now.
Well, yeah, until I do something crazy. Well, I didn't have anything else. I just kind of at this point I kind of wanted to open everything up for questions and then maybe talk a little bit about building that bioreactor.
Hello?
Yeah.
Okay, cool. Is my microphone like fucked up or anything?
No, you actually sound okay this time.
All right, sweet. So as far as the actual production of a SCOBY for the bioreactor, I think that it's definitely a good idea if you were able to control like the secretion. The problem is that like obviously you want to separate the organism from the products that it's producing, but in this case you have to have a method for triggering the different layer formations. So like you have to have something for triggering the argonate synthesis, but then you have to trigger the silk on top of that at the same time. So it's like that's one of the main issues, which is why they did it in multiple steps before, so that they could control those levels. That's really the only problem that I see with it. If you could do that, you kind of have to figure out which different proteins that it secretes naturally that control those functions. But that would be a whole other research phase in itself.
Yeah, I was thinking about that as well. I don't think you're wrong in going down that line. And I always go back to how does the organism actually do this? I suspect I have no indication that this idea is the case, but I suspect it might have something to do with circadian rhythms or possibly temperature changes, which would, equate to, like a circadian rhythm of the ocean, right?
Yeah. Yeah, there's a, interestingly enough, I'm applying to a lab that works basically on sequencing the genomes of a lot of just like ocean-derived organisms. So, for example, one of the things that their research group found is that the majority of interesting products that you're going to get from most of the marine life is actually synthesized in multiple steps scattered throughout different genomes, or not different genomes, but different chromosomes. So it's kind of hard to trace exactly what's causing what, which is why there's like an entire field dedicated to it. So that would be one of the big like stepping stones, because they struggle to even synthesize like basic stuff because they're doing it for like drug purposes, right? And in this case, the way that I imagine it happening in my head is a combination of a bunch of different proteins that are all working together to form that weird ass matrix. So that's probably why I advocate for a more physical approach, where we're just kind of combining the two products together rather than trying to engineer something from the ground up, because I don't know if that's physically possible. But I'm still open to everything, I guess.
Yeah,
Thoughts?
the paper that I referenced, did that change? Yeah, the paper that I referenced, I don't know if the slide updated or not. The paper that I referenced here, the mollusk shell formation, new concepts for understanding biomineralization process. They talk about the two different kinds of protein. There's the silk type protein and was it beta chitin, I think.
Yeah, chitin B.
Chitin B. Okay. Um, and those come out at different times, right? They get secreted at different times, and I guess that's what forms the matrix, and gives it substance.
Yeah, the main issue is the like it's called like the nanosperities. And in this case, I don't know the exact reference, but it's like the part where the crystallization happens. So in the paper that we reviewed, I think like 2 weeks ago, it basically showed that you could force it to crystallize in like a pattern that was similar to nacre, but not exactly what we wanted if you did it by bacterial deposition, which is like the thing that we use with the urea bacteria. But even the problem with that is like it's similar, so we could probably get something similar enough. Um, but like even when we're talking about like silk secretion, in the case of the other paper, they basically did the same thing except they just used a different binder instead of silk. So I guess we could use something that is more similar to mollusks, because there haven't actually been a lot of studies trying to simulate this bacterially. There are only like 2 that I've read of. So you'd have to do a bunch of different trials, and you'd probably have to like slap some genes out of some of the bacteria, but it could do something, I guess.
Just had a crazy stupid thought.
Sai, your screen froze once more. Can you just kill the video stream for now? And we'll just do this over voice. Thank you.
What's your crazy idea?
Oh, if you just wanted a key for your keyboard, you could do a little operation on the shell of a mollusk and shove a key in there.
Right.
And wait a number of months.
This is more of a proof of concept about making forms,
Yes,
as
I know.
opposed to, yeah. If I just wanted a key for my keyboard, I could take a Dremel and make it out of wood. I could, I don't know, make it out of the foreskin of 1,000 babies. Whatever.
Well, in the problem with the making it like shoving it into a mollusk is like the problem that people have with making synthetic pearls anyways. Like people always talk about synthetic pearls being weaker than natural pearls because they're made by basically shoving a plastic bead into the mollusk to make it irritated until it coats it in nacre. So it only has a not great coat. So like it would be coated in it, but it wouldn't actually have any of the good material properties that come with not great itself. So yeah.
Yeah. So, but we're not going to be capable of making the exact same thing. Like, we just don't have that sweet protein cross-linking goodness. But we can make something similar. And again, this is more about proof of concept than it is about perfect duplication.
Well, yeah, but I mean, if you wanted to just like code it, then you could just do what they do with synthetic pearls anyway, which kind of defeats the concept of making a bioreactor to begin with.
Do you think that you could take a keyboard key and stuff it inside of an abalone and then have it make a coating around it that had any sort of shape that would be functional whatsoever?
No, but I could take a bunch of dock grade, grind it up, and refunctionalize it like they do with a lot of other synthetic pearls. So like, it would look shiny, but
It's not about the shiny, it's about the binding properties.
which is why it would be better to have a full key made out of it, because otherwise I mean, I guess, I don't know.
Right. That's why we're trying to make a full key. This is like when we tried to make the sunglasses.
You made sunglasses?
Kaido glasses, yeah. We tried to make a pair of sunglasses out of chitosan, but they were floppy.
They were fragile.
They were pretty sick though. Like if we
Yeah.
if we had milled it out with that like shit that we were gonna send to SpaceX, that would have been awesome.
Yeah, I still haven't taken a drill or a Dremel to that thing.
Yeah, I think if you maybe like if you started going at it and then put it in like sodium hydroxide or something, and just like functionalize the outside of it, then the entire outer coating would be waterproof technically. So that's something to consider as well. But Kaido.
Also, so much chitosan went into that.
Yeah, that was like a fucking bread pan of chitosan that we baked down over several days. It was awesome. But I don't know. I was talking to Justin today as well about making the cup. But as far as making a cup directly from chitosan, the only method that I've seen to work would be the dipping method. It's like the only thing that actually makes sense at this point. But either way, I'm probably getting off topic.
Okay, so I had to completely restart everything because evidently that was my problem. Unplug it and plug it back in. So I kind of missed, hopefully, what was a wrap up on people's thoughts and understanding about the paper. And we just kind of moved back into chitosan because that's where we always end up. Is that what happened here?
Well,
More or less.
More or less, yeah. I did have one question related to the beers, though, because one of my chemistry teachers posed me with the problem of would it be possible to remove the methanol byproducts from the beer to prevent hangovers? Question for anyone.
I mean, isn't that just how we do brewing?
Well, the thing is that like I was reading a bunch of research papers on it, and apparently the thing that causes the hangover the most is the very, very small amounts of trace methanol that are left behind during the brewing process. So if you could find a way to remove most of those at like the very end, then in theory you could have a beer that didn't give you as bad of a hangover.
The amount of methanol that typically gets produced in a batch of beer is very, very small. Typically the way to get rid of that is to heat it up and drive off the methanol, because that comes off before the ethanol. If you get into distillation, it's one of the very first products to come off is the methanol and the Nail polish.
Yeah, the
remover.
head.
The heads, yes.
Then you want the hearts.
Heads, hearts, and tails.
Right.
So you could, if you don't want to like fuck with your beer, you could basically make well, you could use a rope valve so that you're a rotating vaporizer,
Oh yeah.
and you could use one of those to, in theory, pull off the micro amounts of methanol that are in beers. But for all intents and purposes, most people's hangovers come from dehydration, especially if they're drinking beer.
And the other thing too with methanol is the antidote for methanol poisoning is ethanol. So, and there's vastly more ethanol in a beer than there is methanol. So your body takes care of it.
Yeah, this is why hair of the dog is actually a thing.
Yeah.
But you could, in theory, make a perfectly pure beer by using a rope valve.
Well, yeah, I mean, I assume physical methods, you could just distill it out. But I was thinking if there was any way to do it, like from a genetic engineering perspective, but I'm assuming that would probably like defeat the whole brewing purpose to begin with. But
I actually, so this is a kind of, so if you're talking about it that way, no, it's really fucking hard, actually. In fact, you're going to get a variety of different types of ethanol or ethanol with R groups type of thing in your alcohol, which is why they'll suggest that you don't ferment red wine at over 72 degrees Fahrenheit, or and that white wines are distilled at, or not distilled, but fermented at a cooler temperature. It's because even temperature changes will change the metabolic pathways and processes. So, I mean, you really gotta, that would be a lifetime work of selective breeding and genetic engineering to make a yeast that doesn't make methanol. And then on top of it, if you remember the mouth bacteria project, you can't remove one pathway without putting something else in to kind of hold the place.
Right. Else it will be an equilibrium shift. Like
Exactly.
Yeah.
Yeah. So this is just kind of, biology is messy.
The way I'd approach it is on the process side. You're going to get more methanol fermenting fruits than you will grains. Certain kinds of fruits will give you more methanol. So ingredients are going to play a key role. And then fermenting cooler temperatures, I think, is going to give you a cleaner ferment and less likely to throw off some of those crazy things. So that's the route I would go. But to say that you have a fermentation that would perfect.
I think the biohacker route here would be to just modify the liver. That's all I'm saying. Slap a virus in that boy. Give him a lysina liver.
AAC7. I'm telling you. I think that's the liver-directed adenovirus. AAV7. But no. No stabbing each other in the liver. God, people. But yeah, so to kind of scoot this concept sideways, directed evolution, and Sai was originally talking about bioreactors. So let's talk about my favorite type of bioreactor, which is a hyperspecific type, which is called a chemostat. And so chemostats are bioreactors that self-regulate. And so you can make some really cool bioreactors that like have a laser sensor, and so they're constantly measuring the density of the fluid, and so they are feeding and heating and removing and stirring, all based off of this type of thing. So imagine like a flask with all the normal stuff that a bioreactor has, but then it's got an Arduino attached to it that keeps track of it.
These are the ones they typically use for like viral productions in labs, right?
Yeah.
Yeah.
And so, I mean, what you can do is, so Pistachio, you remember working with Koji? So
I still have the Code incubator somewhere.
oh God,
It was
I
so
miss that.
bad.
So Koji has been lovingly handcrafted and bred, like many yeasts have, like wine yeast, all these different types, for like centuries and centuries. But it's only been in the last 100 years that people have made that accessible. Basically, 100 years ago, there were only 10 people that knew how to deal with Koji, which is way different than brewer's yeast, because like that stuff's like everybody's got brewer's yeast. You know, you walk in any house, they're just like, yeah, I'm just yeasting that. So there were only 10 people who knew how to like farm or grow Koji until just about 100 years ago. So it's been opening up very, very slowly. But this is a really, really highly genetically modified organism, and it's just one of those things that takes a tremendous amount of time, because you can't actually know what a modification. You can have a theory or a hypothesis about what a modification does, but because we have such a patchy understanding of biology still, if you just smack something in there and you're like, boom, no methanol yeast, and then you just watch it go completely haywire.
Hey guys,
Yeah.
I gotta go. Our power just went out, so I am off to the data center.
Oh shit. Okay.
Joy.
Yeah.
Talk to you guys later.
Good luck.
Thanks for the presentation.
Hell yeah.
Yeah. Is somebody recording this?
Yeah.
Yes, I think.
Always.
Awesome. Okay.
We have it all.
I'll catch up. I'll catch up on it later.
Cool.
But so there's when you're talking about bacteria and yeast, there's kind of like a middle ground between what we consider to be the very heavy-handed version of genetic modification that we do with larger species, and what we call genetic engineering per se, and it's called directed evolution, where because they replicate so quickly that you can apply pressures to make them express and suppress certain genes. And this is how we've gotten a lot of our strains of yeast that we use for brewing, and also how we got Koji. Now we have access to things like chemostats and bioreactors, and so we can do selective breeding way faster. I'm sorry, that is the punchline. Somebody else is supposed to contribute now.
Yeah, I'm looking at chemostats. So yeah.
The University of Washington, if you type in University of Washington chemostat, there is a 3D printed chemostat
Wow.
that they put together, and it's, for me, it feels, well, one, I get to work a little bit on a project with that, but two, I actually think it's a really great example of the type of stuff that people like us can build, and bioreactors don't need to be this gigantic monstrosity made out of, like, look at these bolts. This is monstrous. Like, wow. You know, they
I
can
don't know. It could be a -80 turned on its side.
Yeah, it could be a -80 turned on its side with the cedar frame that you built.
That was a good ass incubator. I miss it.
Yeah, me too. But so when you're doing directed evolution, and especially when you're dealing with bacteria and yeast, you don't actually need these large systems. because you're just test bedding. And so you can start working with bioreactors that are the size of, 50 mils.
Saltwater
And
fungus.
yeah, and all of a sudden this starts working. Like then you can just rack them up, and you can do 20 or 30 tests at a time, and you can be like, okay, well, what do I do if I just poke it here? Of course, this requires a lot of like care and attention, but that's research for you.
Yeah, the plastic eating fungus that we did directed evolution on was pretty interesting. Do you still have a sample of that?
You bet I do.
I was always interested as to why it formed that the pink layer on the bottom of the foam. If anybody's interested in what I'm talking about, there was it's P. microspora, right?
That is correct.
Yeah, we got our hands on a sample of P. microspora and then introduced it into a large fish tank after slowly making it saltwater tolerant, and then it started eating away at pieces of plastic that we threw in there. So that was fun.
The goal was to make a plastic-eating fungus that was also capable of living in salt water.
Right. To
Yeah.
degrade those floating islands of
Yeah.
garbage.
Exactly.
Good stuff.
So I think we've kind of like wrapped it up for the paper, right? And Sai is gone, and he was running the paper. I'm gonna say see you guys later. And if you guys want to keep talking, by all means, of course, always. But sorry I missed half of it.
Oh, all you got it recorded. So, but yeah, I'm gonna look up all the terms I didn't know about. It was awesome.
Do we know what next week's paper is going to be?
Currently, that seems to be a little bit up for debate until like literally 3 hours ago. I thought I did know. Now
Yeah.
I hear that there's been some discussion that Keone might be presenting. So this is why. This is why Fast is putting together just a little spreadsheet so that things can get thrown in there and actually organized, because without a calendar app in Discord, maybe there is, I don't know. But without that, organizing a weekly thing with people just being like, hey, 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 and then also DMing each other and being like, hey, 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 is like complete chaos.
It is.
So we're going to make a spreadsheet because that's how you deal with complete chaos.
Where did Keone comment his paper?
That's the thing. I got that in a DM. So.
Damn it, Keone. Okay.
Yeah.
Got it.
Yeah. So, what I thought was happening was that I was going to be presenting again to go back to the structure of how to break down papers. And then I turned that over to Bast. And then the Keone thing happened. And then there's a whole bunch of other scheduling. So we're going to get stuff on the schedule. Don't you folks worry about it. We're going to make
Amazing.
a spreadsheet. Spreadsheets fix everything.
Except broken hearts.
Obviously, you're not good enough at spreadsheets, Pistachio.
I'll try harder. Could fix love with Excel documents if you try hard enough.
Wait, you didn't take that class?
My college wasn't offering it for freshmen.
Well, you are in Florida, so, what are you going to do? All right.
Yeah, by the way, I went to the storage unit and got all of the conversation afterwards, but Pistachio Lab is up and running now.
Yeah, I saw the little picture.
Yep.
If anybody else has little labs that they're setting up. Don't forget that there is a lab design document and a lab design channel where you guys can talk about putting together labs and give each other help about what a lab needs. Because, you know, when we talk about this stuff or we read these papers and then we get really excited and then we're like, I'm going to buy stuff. And it's better to buy stuff for a project or design a lab for a project. So participate in lab design.
Yo.
All right. Bye, guys.
See ya.
Yeah.